afternoon, everybody. I would like to start by thanking the Human Frontier Science Program for awarding me a fellowship to work at the MRC in Cambridge. The MRC I knew 35 years ago, looked like this. And the best and most important place was the canteen, where one had the opportunity to talk to great scientists and discuss with total openness the best of the best that you could do in science. And there I worked with Nigel Unwin, a New Zealand, originally from the island, South Pacific as well, on cryo-electron microscopy of the nicotinic receptor, a neurotransmitter ubiquitously distributed in the nervous system, trying to improve the crystallinity of the specimens that Nigel was using in those days, and particularly using cholesterol as a mediating lipid, a theme that Nigel has followed since. Here you saw him together with Richard Henderson, who got the Nobel Prize subsequently, and honestly, Nigel's contribution was equally important to this subject. Now, what invariably springs to mind when at the mention of the word cholesterol are the negative consequences of this fat for human health: atherosclerosis, cardiovascular disease, stroke, and you go on this type of maladies. Of course, high cholesterol is bad, but the other extreme also appears to be bad. In fact, extremely low cholesterol in blood appears to be a strong risk factor for suicide in persons that have a pre-existing major depressive disorder. Not for everyone, fortunately. Now, in the central nervous system, cholesterol plays an important role in modulating enzymes. And this is the case of the secretases that cleave the amyloid precursor protein, and which generates upon enzymatic cleavage to produce amyloid beta. So increased levels of cholesterol augment beta and gamma secretase activities with a consequent rise in amyloid beta production. So hypercholesterolemia constitutes an important risk factor and a comorbidity for the most common form of dementia, Alzheimer's disease. But on the other hand, on the sunny side of the street, if you like, cholesterol is an essential molecule without which humans and other multicellular organisms would simply not be able to conduct life on planet Earth. Because cholesterol is absolutely necessary to maintain the inside-out polarity of the membrane, and also for the integrity of the cells in terms of functional aspects. The saying goes, it takes two to tango. Here, the shark symbolizes the macromolecule I'm going to talk about, the nicotinic acetylcholine receptor, and the pilot fish that accompany cholesterol, proportions being respected, symbolize cholesterol molecules. So this allegoric image implies specifically a form of symbiosis, more precisely mutualism, where both species benefit from the other from the relationship with the other species. So the first question that comes to mind, I would like to formulate today is, when did this advantageous relationship begin? Millions of years ago, millions of years of phylogenetic evolution have shaped the interactions between membrane proteins and lipids, and in particular, sterols. Sterols have been on planet Earth for a long time and have sculpted this relationship with the membrane proteins to span a wide range of structural and dynamic relationships. In fact, paleobiologists date the appearance of a proto-ion channels about 3.8 eons. Now comes the word in the title more clearly in shape. That is 3,800 million years in American nomenclature. This ancestral protein gave rise to a variety of proteins, ion channels of different shape and size. And the earliest form that appeared on Earth was this pentameric family of channels, illustrated here by Glioblastoma violaceus and Erwinia chrysanthemi ion channels in primitive bacteria, which are structurally very, very similar to the same channels that we have in our brain, in our muscles, and torpedo electric organ in the electric fish. So this relationship dates back to almost 5,000 million years ago. I don't know what happened with my... Okay. I cannot proceed with it. Oh, here it is. Okay. So here you see again, on the left, hopanoids, the most abundant organic molecule on Earth, and its relationship with the ion channel in the primitive bacteria, and on the right side, cholesterol, which appeared only 2.5 thousand million years ago. So hopanoids, this substance that appeared well before cholesterol, observed a function on the membrane of prokaryotes, and this has helped oil engineers to look at hopanoids as a clue to determining the age, the quality, and the thermal maturity of this substance, petroleum, which provided a source of energy which paradoxically appeared on this planet well before we had oxygen. And oxygen, without oxygen, we did not have fire, we did not have combustion. Now, what was the essential, the key element which precipitated the appearance of cholesterol on planet Earth? Well, oxygen. Oxygen started emerging from hydrothermal vents about 2.5 billion years ago, and with it, as we heard this morning, oxygenic photosynthesis. And importantly, some scientists consider that oxygenic photosynthesis is the essential element for the appearance of multicellularity in this planet, and the appearance of eukaryotes, of course. Oxygen is required for the so-called mevalonate cycle, which is a series of reactions in which cholesterol is the end product discovered by Konrad Bloch and Fedor-Lenin, and gave rise to the Nobel Award in 1964. And this was complemented a few years later by the work of Michael Brown and Joseph Goldstein, who received the Nobel Prize for discovering the low-density lipoprotein, which is the receptor for cholesterol in our livers. And what is really remarkable is how these two motifs in the protein and in the lipid-like molecules have subsisted, have remained for so long on Earth. Probably this is the simplest explanation is, if hopanoids function so nicely and so well for prokaryotic organisms, why not keep it for the eukaryotes as well, just modifying a little bit the molecule? And it needs not much of a modification, because as you see here, when you superimpose the two molecules, cholesterol and diplotin, which is a hopanoid, they look very much alike. And this preservation is absolutely remarkable in 3 dimensions and in time. Now, how can you modulate a membrane protein with cholesterol? There are 2 mechanisms that nature has provided to do this job. They exert mutually non-exclusive regulatory functions via indirect or direct mechanisms. In the first of these, it is essentially the physical geometry, the shape of the molecules, such as a cone-like, a cylinder-like, an inverted cone, etc., that provide modulation via physical mechanisms, exerted mainly on the bulk of the membrane bilayer. So if you see here in the synapse, it's not only messenger RNA and proteins that are important. Without lipids, the fusion mechanisms of presynaptic vesicles would not take place, and the receptors would not recycle in the postsynaptic membrane. On the other hand, the direct mechanisms are exerted on specific sites, recognition sites on the membrane protein, especially on the transmembrane region of these receptors, where the lipids exert rapid touch and go type of relationships. Exchange rates of 10 to the minus 8 per second can be measured by electron spin resonance, for instance, in the interaction between sterol-like molecules and nicotinic receptors. Now, direct mechanisms are especially important because they do provide some degree of specificity to this regulation. And 25 years ago, we resorted to in silico measurements, molecular modeling, to learn about the interaction between cholesterol and the receptor, and we ended up with about 15 cholesterol molecules interacting with the transmembrane region of the receptor, in the receptor region that is embedded in the plasma membrane. A number that emerges also from the electron spin resonance experimental data. Now, Grace Brannigan also studied the mechanisms of in silico, and here you see a nicotinic receptor with these green elements. These are the cholesterol molecules in the transmembrane region, and you can see that it's essentially the two phases of the cholesterol molecule, both the rough surface and the smooth surface that interact with, in this case, with one subunit of the receptor. Receptors are pentameric, multimeric proteins, so you multiply this and you get the canonical number close to 15. The real validation of these in silico calculations came only recently by the work of the Hibbs lab in Texas and the Nury lab at the Pasteur Institute, who, using cryo-electron microscopy, they could validate the occurrence of both phospholipids and cholesterol in the transmembrane region of the receptor. They did find these sites and the specific amino acids that interacted with the lipid to our satisfaction, of course. Now, let me show you a couple of examples of how cholesterol modulates function. Let's take this important developmental issue in neurobiology, the neuromuscular junction development. Here you see embryonic muscle cells represented by these cylinders with this monodispersed number of receptor molecules on its surface. In the very narrow time window, only 24 hours between embryonic day 13 and 14, receptors become socialized and become closer together, forming nanoclusters. These nanoclusters have to be distinguished from those that appear after the nerve arrives to the surface of the muscle, a phenomenon that occurs postnatally. This all occurs in the womb of the mother, and it's embryonic type of development. So these nanometer size elements, the nanoclusters, they can be conceived as resulting from a nucleation phenomenon, a physicist would tell you. And these are accompanied, interestingly, for my hypothesis, for cholesterol biosynthetic machinery, changes in this machinery that accompany the development of the clustering process with increases in the production of cholesterol in embryonic muscle cells. So can we image this supramolecular organization? Well, the obstacle preventing direct imaging is something that presumably can be attributed to Ernst Abbe when he was working at the Zeiss factory in Jena. The fundamental law of physics that says that the structures smaller than half the wavelength of light cannot be discerned, cannot be distinguished from one another. This is the so-called the limit of resolution seminal work of Abbe in 1872. Now, there are ways of defeating this barrier, and I will exemplify this by a very close and dear example recently occurring in the Weltmeisterschaft. And this is, if it worked, no, I cannot show it, but the goal, but what I want to imply with this allegoric imaging is that the barrier is still there, and these three gentlemen that have in common being male, carrying German surnames and wearing ties, in addition to these three commonalities, they also share the fact that they are the messes in the world of super resolution by having provided methods to circumvent the barrier. The barrier is a physical law, so it's still there, but they found ways of defeating this barrier. One of these methods is STED, a method invented by Stefan Hell. My young student Javier Bayer and myself visited Göttingen in the early 20s, and this is a microscope, believe it or not. I always ask my students, where do you look at in this microscope? Well, it is somewhere hidden there. Anyway, we can, and we did, image this receptor with this STED microscope, which is a targeted form of microscopy, and we also manipulated via cyclodextrin, enriching or depleting the cholesterol content. We know very precisely how the kinetics of interaction between cyclodextrins and how do they extract cholesterol from membranes. We had to tune this for our cells. Here you see electron microscopy as applied to these cells. There's an optimal concentration, time, and temperature where cells can be depleted of cholesterol without affecting the microvilli and without affecting the morphology. If you go too far in terms of temperature, concentration, etc., you deplete not only cholesterol, you detach the cells from the substrate and they die. But cholesterol depletion has more subtle effects, such as what we found with Jitu Mayer in India, in Bangalore, and subsequently with Virginia Borroni in Buenos Aires, namely modifying the cholesterol content of the cell. You can modify the route by which the cell internalizes receptors from its surface. So the endocytic pathway becomes completely independent of the cytoskeleton of the actin fibers that are the default route, and they become completely different. Phospholipase D starts acting, it's a mess. You can also interrogate a more subtle type of phenomenon modulated by cholesterol by using another single molecule technique like this patch clamp single channel recording. And as you see here, within the relatively limited range of concentrations of cholesterol, you can either prolong the channel mean open time by depleting cholesterol or enrich cholesterol mediated by cyclodextrin enrichment, and either with slow type of recording or rapid type of recording, you can see that it's a sort of fine-tuning of the channel activity depending on the concentration of cholesterol. So cholesterol exerts pleomorphic type of effects on ion channels, and exemplified by the nicotinic receptor, of course. Now, state microscopy, I announced that I was going to show you some examples. Here you see on the left, upper left, the cells that I just show you with electron microscopy. Now with optical microscopy, you can see that they are patched with a high level, a very robust expression of nicotinic receptors, which are these points that you see in panel C, as shown by confocal microscopy. When you switch to STED, you can start resolving these nanoclusters into smaller subunits. Of course, here we are using a secondary sandwich type of labeling with an ATO 532 label secondary antibody, which is a really macroscopic type of probe, and the receptor is only 8 nanometers in diameter. So this poses a very inconvenient to image receptors. In fact, there is another complication named when using antibodies, and it's the fact that the antibodies can cross-link it to each other and form this type of evaginations in the membrane. So, in fact, by using antibodies, we cannot discern between a single molecule, here a 9 nanometer receptor seen on the image, or a group of receptors. Yet, we are in the realm of electron microscopy. As you see here on the right panel, you can see monomeric, oligomeric, and dimeric forms of receptors as imaged with uranyl acetate negatively contrasted specimens. And on the left, what STED is showing us is not too different. Of course, this aggregation induced by antibodies has medical implications. In myasthenia gravis, a devastating disease, autoimmune disease produced by the production of autoantibodies against our receptors, the receptors are clustered by precisely these antibodies. So studying this phenomenon, although doesn't let us discern this fine structure of the receptor, is informative on the mechanism of myasthenia gravis. If we want to go smaller, we can use bungarotoxin, which is a toxin from a snake, a poisonous snake, Bungarus multicinctus, occurring in Taiwan and continental China. We conjugated this with Alexa 647, and here you can see thousands of receptors on the ventral surface of the same cells. The nucleus is relatively spared, and you can see in the periphery small clusters of receptors, and this is using STORM, another technique for single molecule microscopy. Now, today it would be impossible to start studying this in great detail without machine learning approaches. And since in Argentina we are passing through a very difficult period in terms of grants and actually totally lack of subsidies, all you need is a very intelligent mathematicians, and these two individuals here are examples of such that provided enough gray matter to develop these techniques of machine learning, not only for static imaging, but also for single molecule tracking in using super resolution optical microscopy. As you know, artificial intelligence and these kind of machine learning techniques are very pervasive, are invading absolutely every single aspect of modern life. And by looking, searching the literature, I found that machine learning can also be used to look for adulteration of wine, and a wine which has not. I wish I had this were my my own company, but it's not. Sorry. Anyway, we are now resorted to graph neural networks on lyse receptor clusters, and interestingly enough, this has provided us evidence that these cross-linked receptors in the form of self-associated molecular aggregates are not an artifact produced by the antibodies, because Bangalore toxins are non-aggregating ligands, and from that infer the conclusion that self-association molecular aggregates are the biologically important phenomenon that occurs prior to the arrival of the nerve to the neuromuscular, the future neuromuscular junction. In fact, you can imagine that this self-aggregation attract nerve terminals to produce neuromuscular synapses. Now, Schrödinger, at an famous opening lecture at the University Trinity College in Dublin in 1944, formulated this famous question about how physics and chemistry can take into account what happens in a living cell within the boundary of an organism. It's a kind of ante-litteram version of what we are asking today with our interrogation methods, biophysical or biochemical, to interrogate the ensemble population. And particularly, what I'm trying to study nowadays is the dynamics of cholesterol and receptors at the population level. And we can do that with different biophysical microscopy techniques. And the last one of these techniques is MinFlux, the last that we have applied. Why MinFlux? Well, although MinFlux can provide us with insights into very small things, it's not structured information that we are seeking from MinFlux, but rather it excels at following molecules in living cell with unprecedented time resolution. And that is what we have been exploring recently, thanks to a fellowship from the Alexander von Humboldt Stiftung, visiting my friend Christian Egeling in Jena at the Leibniz Institute. And this collaboration enabled us to interrogate in living cells, multiple cells being interrogated in succession at random, following the small range tracking, nanometers in length. And this permitted us, in the course of a few minutes, to accumulate hundreds, and in some occasions thousands, of trajectories with resolutions of 10 to the minus a few hundred microseconds in time scale, and less than 7 nanometers in terms of spatial resolution. And what the picture that emerged from that is a complex coverage of motions that range from the very slow kinetics of diffusion in a plane to a very fast super diffusive motion. So receptors unexpectedly cover the whole range of possibilities in terms of displacements. And what we learn from this is that the behavior from subdiffusive to conventional Brownian motion to superdiffusive motion has to do with the proportion of free tracks to confinement, something very similar to what we saw this morning using MinFlux in kinase in motion along the axon. So this sojourns and free walks, the proportion of this determines the diffusional regime of the ensemble of the population. And we were able to label receptors with Bangalore toxin and use a red polyethylene glycol cholesterol analog to measure by a differential discriminant analysis the tracks of the lipid alone, the cholesterol alone, the toxin label receptor alone, or the two in combination, looking at the two different channels. And in both cases, we could see transient spatial overlap of the two fluorescent molecules in co-tracking experiments, on the one hand, and also applying new methods to analyze motion, we could observe that the receptors covered the whole palette of possibilities, from time-dependent changes from fast to slow and confinement, as I have shown before, to co-diffusion and dimerization of lipid and receptor, to sharing compartments where probably lipid-rich and cholesterol-rich regions of the cell membrane enable a co-diffusion in a more symbiotic, if you like, manner. And just to finish, let me read the conclusions on this part of the talk on the model that best describes the experimental data. It's a two-state ergodic model in which the molecules alternate between free Brownian motion and confinement sojourns in areas of only 36-40 nanometers in diameter and about half a second in duration, which we interpret as arising from obstacle densities, and the obstacles being the receptors themselves. So confinement results from crowding from an homocrowding from the same receptor molecules. And they cover, as I said before, a wide continuum of subdiffusive Brownian on superdiffusive motions. When cholesterol transiently accompanies cholesterol, the receptors, it induces changes in the two-dimensional diffusion, and vice versa. The receptor is trapping cholesterol on its surface, and the two move together. And finally, this is a small group I have had for the last few years, which have accompanied this fantastic journey that was to some extent catalyzed by the Human Frontier Science Program. Thank you very much.